Hi everyone, welcome to the channel Botany with Sadhna. In this video, we will discuss about structure of plasma membrane. Before going to discuss about the structure of plasma membrane, first of all we will discuss about a brief discuss about the cell. So, cell is an aqueous compartment surrounded by cell membrane which is capable of independent existence and perform essential functions. And as we all know that cell is the basic and fundamental unit of life. And the first as we all know that the cell was first seen by Robert Hooke in 1665, where he saw in cell in cork cell where he only actually, and that is a dead cell, and he see only cell wall, and the living cell was seen by Anton van Leeuwenhoek. And then we will discuss about some theories of cell theories. Sheldon and Chevan independently gave these cell theory, in which they describe about that all living organisms are made up of cells. Later, Rudolf Virchow came and he gave an extension to their cell theory, in which he says that life came from pre-existing cells, means cells which which are arise from pre-existing life or cells. Now we will the cells on the basis of their structure is of two types: prokaryotic cells and eukaryotic cells. Prokaryotic cells are the mostly unicellular cells, whereas eukaryotic cells are multicellular cells, but they can also be unicellular. Prokaryotic cells are also present. So the main difference lies where in eukaryotic cells, nucleus is surrounded by a membrane called as nuclear membrane, whereas in prokaryotic cells it is absent and nucleus is present in the cytoplasm. And different organelles are also present in eukaryotic cells, and most of the organelles are uh, surrounded by membrane, whereas in prokaryotic cells membrane bound organelles are absent. So now we will discuss about the structure of plasma membrane. So first of all, what is plasma membrane? Plasma membrane is a and fluid dynamic fluid structure surrounding the cell, and it is the external boundary of the cell which regulates the entry or exit, or we can say that molecular trafficking of the cell. And it is selective permeable in nature, means it only allows some of the solutes or or particles to pass through it. So that's why only selective is used, because it allows only some of the solutes to pass through it. And about then the structure of plasma membrane, different models different models came different models were proposed by different scientists and about the structure of plasma membrane, but the most accepted model is fluid mosaic model. It was given by Sanger and Nicholson in 1972. In this model, they says that plasma membrane is made up of lipid and proteins, in which lipid and proteins are arranged in a mosaic arrangement mosaic arrangement in which this membrane is present like this, lipids are present like this, and protein are present in between them, or we can say that here in a mosaic arrangement, and the fluid is called fluid movement is present of lipid and proteins. So this is the fluid mosaic model, about the basics of fluid mosaic model, where mosaic arrangement of lipids and proteins is present and fluid movement of lipid and protein is present. And then in this model, lipids and protein are quasi structure is present of lipids and proteins. The next is chemical constituent of plasma membrane. It is mainly made up of lipids and proteins, and the ratio of lipids and proteins is varies according to the cell type. As we have seen that in RBCs different concentration of lipid or protein is present, or in different different membrane, different plasma different sorry in different plasma membrane of any other cell or any other organism, different composition, or sorry, different ratio of lipids and proteins are present, whereas in carbohydrates are also present in plasma membrane. They can be present or embedded or near the uh, they can be present with proteins or lipids. So when carbohydrates are present with proteins, then they are called as glycoprotein. Glyco and proteins. Glyco is for carbohydrates. When it is present with lipids, then it is called as glycolipids. So the chemical constituent of plasma membrane is having lipids and proteins, and carbohydrates are also present. Then we can call it as glycoprotein or glycolipids. And carbohydrates are mostly abundant in plasma membrane of eukaryotic cells. Abundant in plasma membrane of eukaryotic cells. The next is lipid bilayer form. As I have drawn this diagram, actually it is like this. Lipid bilayer. Lipid bilayer means the plasma membrane is present in a form of bilayer in which a hydrophilic or polar head is present, and two hydrophobic tails are present. And their arrangement is like this. In the plasma membrane, they are present like this, where the hydrophobic tails are protected from the aqueous environment, and the hydrophilic heads are surrounding the external or extracellular compartment, and proteins are present like this on the lipids, on the lipid part. They can be of they can be internal or they can be peripheral. Proteins are of two types: internal protein, integral proteins, or we can say that peripheral proteins. They are classified on the basis of their extraction from the plasma membrane. If by simple procedure or by simple extraction procedure, the peripheral proteins are can be separated out easily, whereas for integral proteins are not easily separated as they are present inside, or we can say that in between the uh, hydrophobic part. So this is little bit difficult to extract the integral protein, and they shows as I told that earlier told that they shows fluid movement, and their arrangement is mosaic type. I hope this is much clear to you.